Hello and welcome to section 1.3 on the scientific method and designing experiments. We're going to look how to design a scientific experiment today and what the components of that would be. And just a quick review, however, regarding the scientific method. If you'll recall, we don't need to necessarily have these in exactly the same order, identifiable steps. However, most of these components are very critical parts of the scientific method and the scientific process. We identify a problem or a question, set up a hypothesis and make prediction about what we think will happen. We make observations, take measurements, perform experiments. We collect the data, compile it, and use logical reasoning to form conclusions and explanations, maybe find an answer, maybe raise more questions. So let's get into what an experiment looks like. So when we talk about setting up an experiment, there are some major components that need to exist in our experiment. We need a problem or a question, something we're asking ourselves, something that can be tested. And when we set up our experiment, we can design a process or a procedure that will test that problem. We also need to include a hypothesis. So a hypothesis is basically an explanation of the problem that can be tested. It's not a random guess. It's not, I think it turns blue. It means that you've looked into the problem a little bit. You've come up with some ideas about what you think might happen and why, and you propose an explanation for that. An explanation stating what you think will happen. The experiment itself will involve a procedure that you carry out. It will be an investigation that is going to test that hypothesis. That's what the experiment does. It tests the hypothesis by collecting data through observations or measurements. A controlled experiment has some very specific things that are characteristic of it. It has a control group and an experimental group. What are those, you might ask? Well, a control group is the standard. It does not get any experimental treatment. The experimental group, on the other hand, is the test. We see what happens when we have this experimental group because it receives experimental treatment. There is something important to remember in all scientific experiments. Only one condition or variable is changed at a time. We never change more than one at a time. A couple more definitions, and then we're going to give some examples of all these things. So I know it's hard just to understand that abstractly, but we have independent variables and dependent variables. So you might ask, what are those? Well, the independent variable affects the outcome. It's the condition that is tested, whereas the dependent variable is the result. It's dependent upon the independent variable. It changes depending upon the independent variable that we've changed. So let's give some examples to help put some ideas to this. Here's an example of an experiment. Let's say we ask ourselves, here's our problem, does caffeine in coffee prevent mold? Good question. I don't know. Why don't you sit and think of a good hypothesis for this question? So you can pause and give your hypothesis. Here's our experimental design. We are going to conduct a controlled experiment. The procedure is as follows. We're going to have beaker 1 over here containing 25 milliliters of caffeinated coffee. And then we're going to have beaker 2 right here containing 25 milliliters of decaffeinated coffee. Both jars are covered. And then the observations of the liquid surfaces are recorded daily for 1 week. I want you to now think and pause the video for just a minute. What in this experiment is the control? What is the independent variable? And what is the dependent variable? So here I filled in just one of many hypotheses. You could have said a lot of different things. I just said caffeine does not prevent mold growth. You could word that as caffeine does prevent mold growth, or caffeine prevents mold growth because the beakers, one doesn't have mold growth and mold growth, and the other one does. And you could have described it that way as well. So you could have said several different things for that hypothesis. Here, the control is the decaffeinated coffee. It's the coffee without caffeine. The independent variable is the caffeinated coffee. It's what we're testing. We're putting that caffeine in there. We're having the same environments in both beakers, but in the independent variable is going to be that caffeine. So the caffeinated coffee is what we're going to test and what we're going to change. The dependent variable is mold growth. It's what occurs based on the experiment, either on the independent variable, the control. It's the outcome, what we see. It depends on the experiment. And are we going to see mold growth or not? The results of this experiment, in our little hypothetical experiment, based on the data collected, the results are that both types of coffee allow mold to grow. So in both of these cups, as we observe, we see mold growing. So was the hypothesis supported by the data? Well, if you'll remember, the hypothesis that I wrote was caffeine does not prevent mold growth. And indeed, the hypothesis was supported because mold grew in both of the beakers. So does this raise new questions? There's certainly some questions that could be going through your mind. Well, could we design a different experiment, another experiment? Absolutely. One thing I want you to keep in mind is that trials are very important. The number of times that the same conditions are repeated. So replicate groups exposed to the same conditions I wrote down there. But we could have done this would have been a much better experiment if we would have had number 1 beakers, maybe 5 of them, and number 2 beakers, maybe 5 of them, to repeat the trial so that we are certain that there is not some anomaly that's occurring that is not explained under the conditions that we have. Now, as we continue on through the scientific method and through our experimental design, one very important thing we need to report our results. Valuable feedback and collaboration occur when we report the results. So in our case, we're not really going out and publishing results or anything, but we're reporting those to our peers, to the teacher, and so we can look at each other's experiments and say, wow, you forgot to do some multiple trials here, obviously, like we didn't do, right? Or here's one idea I have. What if this were happening? What if something else is occurring that you're not thinking of? And that's a really good way to improve our experiments and to improve on them. For scientists, they may write a theory. A theory is a hypothesis that's supported by a large body of evidence. An example of that would be that 1839 the hypothesis that cells are fundamental to life had been floating about for some time, and it actually became a theory because there was so much scientific evidence that was supporting that theory. Sometimes, as you look at the results, as you collaborate, as you report these results, there are new hypotheses that are formed and revised theories and more research that can go on and more tests that can be done, more observations to be made. So it's kind of a cyclical process that continues on as we get more and more data and collect more and more information. Now, hopefully you've learned a little something about how experimental designs are set up, and we're going to be working some more on this in class because it's a lot of information and a lot of ideas, but for an advanced proficiency, I'd like you to just come up with your own ideas. You can, if you want, you can design your own experiment and carry it out and share your results. You can do something else that you have an idea for. So wish you well, and we'll see you in class.